Hello, we are going to take you on a journey with us to see adrenergic agonists from the scope of pharmaceutical chemistry. Sympathomimetics are classified into direct, indirect, and mixed acting agonists. The direct acting agonists bind to and activate alpha and beta receptors, while indirect agonists produce norepinephrine-like actions by stimulating norepinephrine release. And the mixed acting agonists work by direct and indirect action, and will be mentioned later. Another classification for sympathomimetics is being catecholamine or not. The catechol ring is 1,2-dihydroxybenzene. Once there is any change in ring structure or OH position, such as 1,3-dihydroxybenzene instead, it's a non-catechol, and so it will be resistant to metabolism by COMT enzyme, which targets that ring, and hence have a longer duration. Here we can see the phenylethylamine structure of adrenergic agonists, through which we can understand the structure-activity relationship. R1. If R1 is hydrogen, it's norepinephrine, and so targets alpha-1, alpha-2, and beta-1. While when replacing hydrogen with methyl group, it gives epinephrine, which is non-selective and used in anaphylactic shock and cardiac arrest. But when R1 is a group larger than methyl, it's more selective on beta. And if it's tertiary butyl, it becomes more selective on beta-2. While a larger group than butyl gives a compound with alpha antagonistic activity. But here if we come to R2, we can see that methyl or ethyl groups decrease the metabolism by MAO enzyme. Methyl group here gives a compound which is more selective on alpha-2 like methyl dopa, while the ethyl group gives a compound more selective on beta like ethyl norepinephrine. And here's R3. There must be at least one hydroxyl group for hydrogen bonding. If it's a 3,4 disubstituted, it's non-selective agonist. The 4-OH gives a selective beta and the 3-OH gives a selective alpha, while 3,5 disubstitution by OH gives an agonist which is more selective on beta-2 and resists COMT enzyme, hence could be taken orally. Now we'll start talking about alpha receptors. Its activation causes vasoconstriction, relaxation of GIT muscles, and contraction of radial muscles of the iris or mydriasis. Starting from the non-selective agonists as adrenaline and noradrenaline, we can say that the non-selective type causes cardiac stimulation, vasoconstriction, and undergoes rapid metabolism by MAO and COMT. They are used intravenously in hypertension crisis and cardiac arrest, with noradrenaline having more agonist potency than adrenaline. The difference between their structures appears here. As we said before, the R1 is methyl and epinephrine, while the norepinephrine is the demethylated form. Here is the selective alpha-1 agonists. They are used to treat hypertension, bloodshot eyes, and nasal congestion. Here we have phenylephrine, metraminol, and methoxamine. And we can notice that the para-hydroxy group had been removed to give selective alpha-1, as we said before, and hence they all resist metabolism by COMT as they are not catechols. Metraminol is a structural isomer to phenylephrine. And methoxamine undergoes O-dealkylation to hydroxy group for the hydrogen bond to be established with the receptor, and at higher doses of methoxamine it becomes beta blocker. Here the alpha-methyl group in metraminol and methoxamine reduce their metabolism by MAO enzyme. Now, the oral imidazoles are xylometazoline, oxymetazoline, tetrahydrozoline, and nafazoline. The difference appears here, and we can see the main difference between xylometazoline and oxymetazoline, which is a hydroxyl group in oxymetazoline. And here's the tetrahydrobenzene ring in tetrahydrozoline, while nafazoline contains a naphthalene ring. They are used topically in decongestants as nasal and in eye preparations. Now we came to the end of our journey. Hope you could enjoy it. Thank you for listening.